just as a preference, there's can only be 20 players in this Kahoot. So if you have friends in this room, please share with as many people as you reasonably can, because I don't know, Kahoot is just wacky like that today. So I'm gonna start it up. Oh, let me start. Oh, never mind. Oh, that was weird. I guess more than 20 people can join. I don't know what just happened. Just see if you can join. If you can join, just join. We'll see what happens. Okay. We haven't really started yet. Yeah. All right. If everyone's ready, we can start. All right. Let's begin. I'm just gonna run through the Kahoot so we have time to set up. Yeah. If you can see the structure. Unfortunate. Okay, okay. Can you see this? Can you see this? All right, let's just get rid of it.
Yes.
There's no show media. Okay. What? There's just, you want to come over here? Oh, this?
Yeah,
oh,
there
you're
we
so smart. Okay, sorry. And so when we look at AGAD, right, like, It's the same concept. Like, what is A next to, right? So that hypothetical amino acid would be right next to that carbon ring, right? And so we're talking again about like hydrophobicity. And when we look at our options, right, like which ones would be nonpolar? We have isoleucine, serine, aspartic acid, and arginine. Well, like you don't have to memorize not the chart, right? But you should recognize that isoleucine only has CH bonds, or CC CH bonds. Very nonpolar. Serine has an OH group. Arginine is positively charged. Aspartic acid is negatively charged. Yeah. So more people outside. Yeah. We were safe. We were safe. All right. All right, like when we look at the interactions, B would make, well, it's an NH group sticking out from the dopamine. So like, well, it's probably a hydrogen bond. That's the most likely interaction. So as also amino acid, the one that's most likely to make a hydrogen bond is that tyrosine. Okay, that's pretty good, right? Like the most likely interaction in this case, right? The NH2 is positively charged. You're probably looking at an ionic interaction, so it's either with glutamic acid or aspartic acid. Just make sure you don't get tripped up with glutamine. Glutamine is a separate amino acid. Hopefully you guys remember this because this is on one of the problem sets. All right, seems good. All right, like just because two proteins have the same domain does not mean that the whole protein are like identical, right? Just a portion of the protein is the same. So there's a lot to unpack, but just relax, take a deep breath, and just read what the words mean. Oh, that's wonderful. Okay. So, you know, most of us got it, but there's like a chunk who didn't. And right, when you're looking at this structure, what you're really looking at is not. Can I show it now? Okay, sorry. Right, like when we look at the picture, the diagram, and we're reading these words, the word we're looking for is monomer, right? So monomers make up a larger structure, and proteins, each monomer is essentially a different polypeptide, and each polypeptide will be a subunit. So there's monomer A, monomer B. I see where you're getting the 3 right? Like there's maybe 3 different colors, but that doesn't really matter. That's a different thing they're trying to analyze. Yeah, so if you just read the picture, you'll read A, B, C domain. There's 3. And like these kinds of questions are representative of what Laura might give, right? Like it's not, it's a diagram and she won't explain it, but you have to piece together what each element means together. Yeah, excellent. That's pretty close. Right, like when we're breaking down a question like this, you're trying to piece out what the pertinent information is, right? And so, right, if you're getting bogged down by the PP, ADP, like don't think about that too strongly because like that's not what we're learning, right? Details that you don't super recognize on a test, like don't let that distract you from what you do recognize. And so what you should have recognized is really the second paragraph, right? It's regulated through phosphorylation and dephosphorylation. Those are post-translational modifications. And then the second part, FBP, who knows what that is? Doesn't really matter. It says it binds to a binding pocket far away from the active site. What does that sound like? Anyone? Anyone? All right, it's fine. We get it. It's late. Well, that's allostery, right? Like allostery is when you bind to one site on the protein. That's not necessarily the active site, but it affects the active site in a way. So it's allostery and post-translational. Like there's no nucleotides really. That's one of the substrates, but that's not part of its regulation.
You're right, Robbie. You are cool.
Excellent. Ah, seems good. Right, and so it has the ER retention sequence, not the ER localization sequence. Without that localization sequence, you're not inducted into the endomembrane system. And if you don't enter through the ER, you're not in there anywhere. Right, that localization sequence is the NLS in this case. Does that make sense, everyone? Okay.
Sorry.
So there's gonna be a diagram. All right, so let's break this question down a little bit. So, right, if RAND gap started not functioning, it would mean that RAND GTP is not being converted into RAND GDP in the cytosol. And that is the key step that allows exportin to be released from the ternary complex and for the cargo protein to be released. So that's like why the yellow option is the answer, but let's look at this red option, right? If RAND GTP cannot let go of the ternary complex, right? Exportin cannot let go from the ternary complex either. They're all stuck together in the cytosol. And exportin can't, right? Like if we look in the cycle, once exportin is free, it's being re-imported into the nucleus. If you are not letting go of that ternary complex, then exportin cannot be re-imported into the nucleus. There's going to be an accumulation of exportin in the cytosol. All right. I'll let them see. Okay. Okay. This would be the role of RAND GAF, right? GAF exchanges GDP for GTP. Gaps hydrolyze. They make a gap in the GTP into GDP. Good. So, right, ubiquitination is a post-translational modification. So, like, I understand why you might think that's protein activity control. But when we think back, right, to the heat shock response, to the UPR response, what does ubiquitination actually do? When you're tagged with ubiquitin, you're marked for destruction in a proteasome and you get destroyed. Like you're not regulated in activity really, you just die. So there you go. All right, she talked about this in lecture 4 of week 3. If you don't remember Peabody's, rewatch that video. She talked about it for like 30 seconds. Basically, this is like the only example she'll give of mRNA localization. So on the exam, right, you might have a question where it's like, give an example of this, and she might say mRNA localization control. What were you gonna say? All right. You have to have all the examples of each type of control, at least have one going into this exam for every single type of control. Yeah. She says in the lecture videos, like, yeah, like you don't have to understand really how it works, but just know this as an example.
It'd definitely be something good to write down on your sheet.
Yeah, sure. Yeah, so prokaryotes don't really do RNA processing. Like they don't engage in like the 5' capping, poly A tail, RNA splicing in the same way eukaryotes do. All right, and then okay, Rue. Dave. All right, congratulations, Ashman. Give a slideshow we're gonna do.
Okay, so the question was, what would happen if you had a non-functional gap if there wasn't?
Can you guys hear her?
No.
The chat. Okay, just I think you have to stay right there. I know.
Okay, people, we're answering the question if we didn't have a gap or if we had a non-functional gap. I don't care for this video. So we're looking at, in the case of gap, we are looking at import or export. Anyone? We're looking at export for the most part for gap. Well, Yeah, only export. So gap is found in the cytosol. Gap in this is right, can you see the mouse? Gap is right here. So this is the step we're looking at for gap. So run down quickly, gap basically binds RAN GTP. So that's RAN in its active state that is bound to important. And it changes RAN's conformation, and that causes it to speed up its hydrolyzation. So RAN is a enzyme, but it's a really bad enzyme. So it hydrolyzes GTP, but it's not very good at it. So GAP essentially comes in and helps speed up that the hydrolyzation process. So if we were to not have GAP or having non-functional GAP, you can imagine that we can't switch RAN GTP then to RAN GDP, or it's just going to take a very long time. So if we never change this conformation, or we do it at an extremely slow rate, important is going to stay bound to RAN GTP. It's actually not important high export really. It's going to stay bound, and if we don't have any important, because it's all bound, then our cargo protein can never bind important, and it will never go into the cell. So if we have a non-functional or no gap, we're going to have decreased or no import. Does that make sense? Okay, head nods are great because I'm going to keep explaining it and beat the dead horse. Okay, good. Okay. So then we have the same question about GEF. Like I said, these are great questions because this is essentially what you need to know and be able to understand for the exam. So GEF is up here. This has to do with export. And GEF's function is to bind RAN GDP and change its conformation so that GDP dissociates out of the pocket that is bound in with RAN, right? So it's important to know that we're not turning GDP into GTP. GDP is floating away, and GTP is in very high concentrations in the nucleus. It is only in the nucleus. So it's going to come in really fast, bind RAN, that changes the conformation, activates it, so then it can bind exportin, which then can bind its cargo protein. If we have no GEF, or we have a loss of function GEF, then we will not be able to bind exportin, not be able to bind our cargo, and we will not have export, or we'll have very little export. Does that make sense? Yes, good. Okay, what's my other RAN gap? Oh, cytosol. Okay, someone said cytosol. So this question essentially is if GAP and GEF were switched. Oh, yeah. It's going to accumulate. I mean, if it's supposed to go in the nucleus, it's going to accumulate because it can't go in the nucleus. So GAP, right? It's not, it's already bound. It's not letting go is the issue. So it's going to stay bound to important. Why is it doing that? Oh, GEF. If GEF isn't there, then we're going to accumulate in the nucleus because we can't. Do you know why it's doing that? Is it the connection? So exportin and the cargo protein are in the nucleus. They have NLSs, I assume. Like that's how exportin gets in a retention sequence. So it can't get out without binding RAN GTP. So if we don't have GEF to make RAN GTP, the active form, then it's just gonna accumulate in the nucleus. It's never gonna bind and it's gonna accumulate. Does that make sense? Okay. So then there was a question about if we switch GEF and GAP, and put GEF in the cytosol and put GAP in the nucleus. So if you think about it, that's a hard question because it depends. If GEF is not in the nucleus at all and it's only in the cytosol, then export's still not going to happen. And same with GAP. If GAP is in the nucleus and not in the cytosol, import's not going to happen. And it's the same reason, it's the same mechanism that we just went through. Yeah, so just understand that mechanism. All right, let's do another question, Robert. What's another question for me? Okay, let's do something else. Okay, good.
Mhm.
So the next question is there a priority of signal sequences? Like if the protein has an ER signal sequence and a mitochondrial one, where will it go first? So we touched on this in class. We're just gonna do another quick rundown of it, and Gabe's gonna take this one.
Yeah, so. Yeah, so there is an order of priority for the signal sequences. Yeah, so the very first priority would be the priorities go co-translationally read and then read like post-translationally. And so there's some other orders that like go through like, or that depend on like where it is in the endomembrane system if it has gone there. So the very first like highest priority is the ER localization sequence, which is read co-translationally, which means like at the very beginning of translation in the cytosol, as soon as the signal sequence has been translated from mRNA to protein to the amino acid signal sequence, a protein comes by and binds to the sequence and brings the whole complex so that it can be continue to be translated. When it
Let me
is
just pull
in,
something up.
to translate the rest of the protein into the ER. And if there are signal sequences, such as like a mitochondrial one, or a nuclear signal sequence, whether NLS or NES, like import or export sequences, it doesn't matter. If there's a localization ER localization sequence, then those aren't going to be read, because once it enters the endomembrane system, the proteins that can recognize the signal for those other ones, like the mitochondrial or nucleus ones, are not going to be there. They're not going to bind to them. Like once it goes into the endomembrane system, it's either going to go all the way through out to the end of like to be secreted out of the cell, or it's going to stay in one of the spots within the endomembrane system, within one of the organelles in the endomembrane system. So somebody had asked if a protein has an ER signal sequence and a mitochondrial one.
Answer that chat question first.
Okay.
That goes back to this question. Yeah. If we don't have import or export, then we can't import. If we don't have import, then we can't export. If we don't have export, we can repeat that.
All right, so Sadaf, important and exportant are required for, import and export in their respective cycles. Like that's what allows them to enter and exit the nucleus respectively.
I just want to mention that import and export aren't like equally, like we're not importing and exporting. It's not like a cycle necessarily. Like sometimes we import cargo in and it like stays in. We recycle our RAN and gap and gaps stay where they are, but like the RAN goes in and out, but it's not like a one-to-one thing, like we're going out and in and out in, So some things need to go in, some things need to go out. So while all of it's kind of involved, it's important to recognize like you can still have functional export even if you don't have import, and you can still have functional export if you don't have import. So it's just important to think like, yes, it's kind of a cycle, but at the same time, they're happening independently of each other. Okay, that's all I want to say.
Somebody asked if a nuclear signal sequence and ER sequence with retention, which pathway takes priority. Okay, so for this one, I think I just explained that, but yeah, like if it had all three of those, then like a localization, ER localization sequence and a retention and nuclear signal sequence, it would end up in the ER just because the localization sequence had already been read co-translationally, and so the protein had been folded or it had been translated into the ER, and then it would already be in there. It can't leave. And then somebody else asked if the NLS or NES sequences are closer to the N terminus, and would it matter? It doesn't really matter where they are. It's not about like where it is in the polypeptide sequence. It's more about where it is in like the folded structure of the protein itself. And it just needs to be, I believe, on the outside.
I think they're.
It's right
asking
here. Yeah.
If you have both, yeah.
Okay, oh, so the other thing is like, if you gotta like think about it like logically, like these proteins are being made in the cytosol outside of the nucleus. So like if it has both though, it does, like it will be imported, right? Because like there is like important will like bind to it when it's in the cytosol and bring it in. And then once it's in there, if it had both, then it can be exported back into the cytosol and like can repeat in a cycle of like import and export. So it doesn't matter like if it's closer to the N terminus, it just matters that they're like where they are in the tertiary structure, and they can both be read.
Okay, and then the rest of these are not specific. So we're gonna go back into some earlier material. So detergents, we went over in class. I'll go over quickly. We did a good discussion about this. People are confused about what the definition of a detergent is. Stop thinking like laundry detergent. All you need to know is that a detergent comes in and basically takes the place of the interactions that are happening. So when you're dirty or your clothes are dirty, think of your laundry detergent. The reason we're clearing the dirt is because the dirt is non-covalently bonded to your clothes. The detergent can come in, replace those bonds, and that dirt goes away because it's no longer bonded. And then we can rinse it with water. So that's all you really need to know about detergents and why that messes up interactions. Do the nonpolar side chains that form the hydrophobic core in a folded protein experience van der Waals bonds? Yeah, that's all nonpolar bonds do. If they're in the core, that's how they're interacting with each other. What aspects of prokaryotic transcription differ from eukaryotic transcription? And which parts are important to class? Well, if we talked about it, it's important to class. I'll start with that. I want like more
Processing, I
detail.
think.
Huh?
Mostly just processing.
Yeah, so the transcription, the biggest difference we talked about was in processing, not transcription necessarily. Transcription, it's the difference between a sigma factor in a prokaryote and a transcription factors in eukaryotes. And then we talked about how the promoter region, the transcription factors can bind to a lot of different regions in eukaryotes and folds. We talked about that today versus prokaryotes. There's just one location and they have to be close. If someone wants to go to the mechanisms, that's different, but I'm not going to answer a question no one asked. Please go over protein structures and folding. I'm going to come back to that because it's a long question. That's folding. Domain versus subunit, any analogies? So a subunit is like if you have a bunch of train cars to make a whole train, each car is a subunit versus a domain. Say that you have someone on each train car that like, I don't know, like takes and punches your ticket. They all have the same purpose and some cars have it and some cars don't. That person would be a domain. So they're in different parts of different proteins and they all do the same thing. Versus a subunit is just a part of a protein. Okay, so protein structure and folding. So protein structure and folding, we have 3 levels. We have primary, which is your amino acid sequence. We have secondary, which is your backbone, alpha helices and beta sheets, and that's hydrogen bonds. And we have our tertiary, which are how R groups interact with each other, which are your hydrogen bonds, van der Waals interactions, ionic, we have disulfide bonds, ion dipole interactions. So pretty much all of those. The only one that's covalent are your disulfide bonds, and they can only happen between cysteines. That's important for folding. Quaternary, you have all the same examples of interactions. They're just between 2 different polypeptides. Yes, question? Yeah. Yeah.
Yeah, so the question is for the Zoom. There's the example that we did with domains and subunits with one person versus two people. One person would be one subunit and the two people would be a protein with two subunits, and their hands and feet would be the domains. So I'm going to simplify and just say hands because hands and feet are different to me. So there's 6 hands. We'd say that's one domain found 6 different places. So it's found 4 times in one protein, but it's also found twice in a different protein. Domains have the same function throughout different proteins. If they have the same function, they have to have similar or the same structure, because structure leads to function. So domains and subunits aren't actually related at all to each other. Like they're 2 very separate things. And just because you have more than one subunit doesn't mean you'll have a specific domain. I know people get really mixed up on that. Does that help? The hands. I really like that. Thank you for bringing that up. I'm going to start using that. So yeah, so that's your structures and folding. It just says go over protein structure and folding. So if that person is on Zoom or in the room, could you ask like a specific question? How long does the test usually take? The test, people can finish in an hour and 20 minutes, or they can take 5 hours. It really depends on how fast you write and how long it takes you to answer questions.
Let's say 2.5 hours. Maybe like 2 hours.
I'm a really fast writer, so I would finish in like an hour and a half and leave. But we were here for the final for 5.5 hours, just because some people really like to check their work. And we have police day, so it's up to your pace. The questions are very similar to problem set, so yeah. It is all open response questions, which is a blessing because the reason I almost failed the first exam in this class was because of the multiple choice. Did you have a question? Okay. Do we have any other questions in here? I don't want this to take 45 minutes, but we had like kind of a Kahoot and wanted to like have people ask questions. I thought there'd be more in the forum. We can go through other topics. Does anyone over the Zoom or in person here have questions, topics that they just want me to cover broadly? Yeah. Yeah, so if we're talking about ubiquitination, you don't need to know the specific process. So the question for my Zoomers, I can't believe I just said that. This is going on YouTube. For people on Zoom, the question was for ubiquitination, for it to be targeted for degradation, we need 4 ubiquitins. When we talk about ubiquitination, we're just assuming that it has everything it needs for that process. You don't need to know the particulars. Does that help? Okay. Any problem set questions from problem set 3 that we weren't sure on?
Yeah. So I remember talking last time, there still has like RNA processing. Yeah. Because it all takes place like in the cell itself. Like there's no.
Oh my gosh.
It's like for the purpose of this class, like when we're talking about eukaryotes, like the difference between them, like eukaryotes have this distinction between like, or they have a distinction between like translation and they have to control. But yes, for the purpose of our class, it's like no RNA.
So when they're asking about the part, so can you review the types of regulation? Yes. So
Transcriptional control we'll start with, from DNA to RNA. Please shout out some transcriptional controls. I'll wait. Transcription factors, we need transcription factors. What else? Chromatin compaction around your histones. If your chromatin's super tightly wound, bless you, we can't transcribe it. If it's looser, then we can transcribe it. That's two great examples. You're probably not going to need to know more than two for each. RNA processing control. This is kind of what we were just talking about, the difference between eukaryotes and prokaryotes. So for eukaryotes, what's some RNA processing control examples? Poly-A tail. There's like 30 of you here.
What's on the other side of the 3'?
Yeah, we got the 5' cap. We got, what's our favorite thing that differs between prokaryotes and eukaryotes? Splicing, introns and exons. So we got splicing. So those are all RNA processing control, and that is how eukaryotes, that's a big place where they differ from prokaryotes. So mRNA, we get to the cytosol, we have mRNA transport and localization control. What's something we talked about for this one? I would know this one because this was on the exam last year and no one knew it. P-body. You don't need to know much, but we need to know we can send it to the P-body. What sends it to the P-body? Anyone? mRNA, right? Huh? Oh, never mind. That's week 4. No. I think we talked about it more last year. Don't worry about it. P-body, great example. Know that. mRNA degradation control. This was what I was thinking of. mRNA degradation control. How do we degrade mRNA?
Isn't your problem set? Ubiquitin? Well, right, like what is being ubiquitin like? Is it mRNA or protein? Oh, sorry. It's all right.
This is another tricky one.
Okay, so I know we didn't go over in
You
the class or the lectures super well. This is like a week 4 topic, but right, it was in your problem sets. It should have said like siRNA based risk, right? Do you guys remember that from problem set? So that's called RNA interference, and that is a mechanism that eukaryotes use to target mRNA that it doesn't want. So I don't worry about it too much, right? So just write down like siRNA, miRNA based mRNA degradation. So yeah, so just don't worry about it too much right now. We'll go over it more in week 4, and then you'll understand like exactly the mechanism of how it works. But all you need to know is that it can degrade mRNA.
Translational control. What do we need to translate? We need access to the ribosomes. Someone asked a question. What does the P-body do? We talked about this in the Kahoot. Robert, you want to explain this again? You explained it much better than I do. The P-body.
So P-bodies are localizations in the cell. So, right, you understand like cells have organelles because organelles provide structure to the cell, like it allows for specialization. So P-bodies are not like formal organelles in that they have like a membrane or whatever. It's really just like a place in the cell that because of other proteins, maybe we shouldn't go into much detail. Just think of it like a closet space in the cell, right? Whatever mRNA the cell doesn't want actively in transcription, it's just going to throw that mRNA into this like closet P-body where it can't interact with ribosomes, right? And so that localization is regulating transcription levels, because if the mRNA is not touching the ribosomes, no transcriptions, no translation, I'm sorry, no translation can occur. Hope that's okay.
Okay, protein activity control. This is great. We did this with important and export. What's some stuff we do to control the protein's activity? We can stick on phosphates, methyls, different groups to turn on and off. That's what happens, GTP versus GDP. One has an extra phosphate. We combined it to other things, inhibitors, activators. And then I'm going to finish this and then I'm going to answer that question. Protein degradation control, we talked about this, ubiquitination. When you stick a ubiquitin, it's ubiquitin mediated proteolysis. Okay. Is mRNA transport and localization control. Oh, it's mRNA transport and localization control. Oh yeah, the P-body. Okay, sorry, I'm not. The P-body is mRNA transport and localization control. Okay, and then, do you have any other questions? How do you go through gene regulation problems?
Think about what's being targeted. That's like my best advice. Like when you're reading.
They can't hear you over here.
For gene regulation problems is like read what the initial thing in your problem is being targeted. So when I was looking over the problem sets, it'd often be like something targets mRNA or like something what? Phosphorylation of this protein or like right like what are you phosphorylating? What are you attaching these things to? What are you preventing from being translated, right? Like that is that's like the clearest indicator of what type of control, because whatever you're acting on, right, that's what regulation is. Like you're acting on something, that's what's being controlled.
Yeah, so people are asking the translational control was the access to the ribosome. That's maybe one we talked about. And then someone said number 4, the mRNA degradation control. That's the siRNA, miRNA stuff we were talking about. Don't worry too much about that. I thought it was week 3, it's week 4. That's my bad. Okay, any more questions on anything?
Think back on the Kahoots. Which questions did you struggle with? Like those are your gaps in your information. That's what you should be thinking about tonight when you're going back and reviewing your notes. Like go back, like where did I not get the right answer? And we can clear that up right now if there are any more questions about the Kahoot. Yes?
Oh yeah, just go to the top 5 most difficult questions. That's a good one. Go back to that link. Go back to where you were. That's the top 5. And I think the first one is what she's talking about.
This one, all right. Okay, well.
Luddite. Like I told you, I'm a Luddite.
Okay.
We might cure cancer one day, but we're not gonna figure out how to use Kahoot. I'll tell you that much. Okay.
So what was your question about it?
I think I'm not really sure what I was doing. Yeah, so I was not really.
So I think Laura said in her lecture video, it's a little bit outdated, but like the principle of allosteric is really fundamentally like a regulator binding to the protein, not at the active site, like another binding site, which changes the active site, right? And when you change the active site, you alter its function. Does that make sense? And so I think you can go back, like, just think about all the proteins that have regulators, something like, in this example, it was pyruvate kinase. Oh no, those two. Let me just find this, sorry. Where is it? This one? Where's that? Okay. Right, so like when we look, there's an active site and then there's an allosteric site. Right, the binding of the allosteric site to the inhibitor changes the conformation of the active site. So have you ever learned about the Lac operon? Right, like the repressor has a binding site for DNA, but it also has a binding site for allolactose. There's two different binding sites. One is its primary binding site, right? Like the DNA binding site. That's how it does its job. But it has the allolactase binding site because allolactase is the regulator. The binding of allolactose causes the repressors to peel off the DNA. That's like allostery in its essential form. So when we go back to the Kahoot question, right? In this case, FBP is binding to pyruvate kinase, not at the active site. Right? When you hear like binding to a protein at a site other than the active site, that is like allosteric. That's what should pop into your mind. It's something allosteric. Yeah, we'll post a Kahoot somewhere. And then I think someone wants to go over this one again. So, all right, when we look at the structure of these two amino acids, we look at the R group chemistry, right? We look at threonine, it has just an OH group and then like this carbon over here as well, right? So it's mostly the OH. The ratio of like polar parts to non-polar parts is pretty high. It's pretty polar, right? But when we look at tyrosine, right? What is the ratio at? Like there's this OH group, of course, but then there's a lot of C bonds, CH bonds, which are non-polar. So the ratio of polar to non-polar is lower, right? So tyrosine would be more non-polar, and non-polar amino acids tend to be buried within the protein. Is there another question I want to go over? Don't be shy. Like these questions are going to show up on your exam. You want to be prepared. Yeah, Michaela. Okay. So the question for everyone on Zoom was, what would happen if you had the mitochondrial and the nuclear import sequences and that's all you had? And essentially, she probably wouldn't ask that type of question because, right, who knows which one wins out? They're both, they're both acted on after the protein is translated. And sure, like the nuclear one is on the 3-dimensional fold, so maybe that takes longer, but that's not really a question that's easily answered. It doesn't have a root. Like you could argue either way. And so Laura probably wouldn't put a question like that on. She's way more likely to put the ER localization versus the mitochondrial or the nuclear import. Like that's what I'm betting on. So we had another question. Could we go over the drug binding site? Sure. So let's look back over the Kahoot. Hi, Emily. Thanks for helping out. So let's just go over A again. When we look at the binding pockets, right? Just look at what the molecule has. Are you trying to interact with an NH group, an OH group? Like those require hydrogen bonds. Are you trying to interact with CH groups, CC groups? Those require non-polar amino acids. Or are you trying to interact with an ion? So A, it's just a CC bond, CH bond. That requires non-polarity. So when we look at the amino acid choices, isoleucine is the only non-polarity. When we look at site B, it's NH. NH is more like a hydrogen. That's what you should. What creates hydrogen bonds? Well, the answer choices, there's only tyrosine that has that OH group. Like alanine is nonpolar, cystine's nonpolar, lysine a charged amino acid. Like lysine theoretically could create a hydrogen bond, but that's not really what. It's not what it's really made for. Sorry, Amber. Now when we look at part C, we're looking at the NH plus group, which is, you know, a charged ion. It's a positively charged ion. And so what's most likely going to be positively charged amino acid R group. And so that's going to be your glutamic acid and aspartic acid, essentially. Sorry about the mic quality. I don't know how to really fix this. What was A? A was isoleucine because it's trying to bond with like the nonpolar CH bonds, CC bonds, essentially. Let's go over whatever. All right, we already went over everything, essentially. Is there anything else people want to go over, Michaela? No, all you have to say really, like for post translational modifications, right? Like you want to specify that the target is a protein, right? Post translational, it has to be a protein. So phosphorylation is only done to proteins. And then just say like phosphorylation can affect the surface chemistry, like essentially what you said, just say it can affect, like every protein does it a little bit differently, where like there's different mechanisms, and sometimes phosphorylation is activating, sometimes phosphorylation is inhibiting. Like you can't really know unless you know the specific protein you're talking about. Yeah.
The topic notes.
The topic notes.
I
But-
mean, like in terms of like material, I would definitely go through them and like take certain parts like out of them, like maybe try to summarize stuff. Like for example, when it comes to like, I mean, it is like at the very end of the topic 3 or week 3 topic notes, it says that prokaryotes do regulate, and then it gives like a list like in bullet points, and it's just like a few, like it's like 7 bullet points, but it's not like you're writing up paragraphs. So I would definitely like use the topic notes to like, you can like synthesize information from the topic notes, just like pick out like the most important parts. You don't have to like go in depth explaining, like you don't have to like write out like paragraphs, but definitely like there's a lot of good information that you could take straight out of the topic notes, or like paraphrasing it come down. But definitely the topic notes are a great place to look at in terms of like material to write down on your sheet.
Essentially, anything that shows up in the topic notes could show up on your exam. And so you want to use them to guide what you're going to put on your cheat sheet. So I recommend putting on like, oh yeah, sure, it's fine. So like, what kind of things you should be putting on your cheat sheet? It's really like maybe your own diagrams. Like if whatever we showed you earlier didn't really help explain nuclear import dash export, make your own diagram, draw it out again. If you need like specific things, right? Like there's a lot you need to know for this class, but you don't have to know everything. So something like the controls, the examples, maybe you can't memorize every single example. That's what you should be putting down, like the little details that help you explain answers, but which you might not have the best remembrance of. Yeah, it's gonna be one of them. Perfect.
Yes. Nuclear import and export.
Yeah. So like the two main diagrams, you're going to get are amino acids and nuclear import export. Yeah, it's the exact same one. Good.
The ER localization sequence is the one that signals for the mRNA while it's being translated to be brought over to the ER, whereas the retention sequence is the one that after it has entered the ER and is like fully folded, it makes sure that it stays in the ER and does not continue through the rest of the endomembrane system.
Yeah, so the question is, the only localization and retention sequences. And so retention sequences are only for the endomembrane system.
Yeah, it's only needed because by default, it will like go through the whole endomembrane system and exit the cell if it doesn't have that. Any sequence, a good way to remember is like it's a good way to remember is like it will automatically like go out. So any sequence that like is called like retention is going to make sure that it's retained at that point or at that organelle within the endomembrane system. Yeah, I would also like draw like this diagram right here, or at least just like, I mean, it'll take a lot of space on your sheet to do that, but you could at least just like write down like the steps in bullet points. But I definitely wouldn't leave like any space like wasted on your sheet, or not like a whole lot at least. Make sure you like fill it up completely, because like you are given or allowed like a sheet of paper front and back, so like take advantage of that. Yes. Why I would go to the ER? Yeah. And not the mitochondria. Yeah. Because when the protein is being translated, like if you think about like the mRNA, like is like bound to like a ribosome, the ribosome starts like churning out, translating the sequence, or like reading the mRNA sequence and producing, a sequence of amino acids. Once like the amino acids for the ER localization sequence are produced, then another protein can come by and grab onto that. And like when a protein has an ER localization sequence, it's typically at the front, like at the end terminus. So like as soon as like the first bit of the protein is translated, it will like immediately have a protein bind to it and bring it to the ER. And so it won't continue, like the mitochondrial signal sequence will be like later on in the mRNA, but it won't be translated until it has already been brought to the ER, the surface of the ER. And so when that happens, right, like the mRNA or the mRNA that's signals or codes for the signal sequence for the mitochondrial localization, it won't like be translated until it is literally in the ER. And at that point, it can't be read to go to the mitochondria. Like there's no proteins in the ER that have that function of like recognizing that sequence, binding to it, and bringing it outside. The only time a protein would ever go outside of the ER back out is for degradation. Does that make sense? Mm-hmm. Anyone else have questions or like that? Yes.
Okay.
It is really confusing. The whole like equation.
Like KD is equal to PL over PL. Like,
Yeah.
I-
So what I would do is
I think so.
you guys remember that there's like a fill in the blanks question on the problem sets about KD? Okay, well, anyways, I would basically the main thing to remember is just like the inverse, like how when KD is lower, the affinity between two proteins is higher, and the inverse is true. The higher the KD, the lower the affinity. And it's just good to know that in terms of like a protein and the reagent, and then it's like reactant. Like when a protein needs to, when an enzyme needs to like catalyze a reaction, you want the protein that initially binds to it to have good affinity for it. But like once the reaction's already taken place, you want it to have like low affinity for it, therefore high KD, right? And at the beginning, you want to have high affinity, low KD. And I know there's like this statement about like if like when the KD is like half of this, then the ligand, whatever. Like personally, I don't remember that, but if you just go to the topic notes, there's like a little blurb there about it, like a couple of sentences. If that does come up on the test, like you needing to know like that little blurb about it, you can literally just write the sentences on your sheet and then just type or like just write it like on the exam when it comes time for that. I mean,
Like the way she
Not really,
expects you
no.
to understand KD is she just wants you to understand it's the opposite of affinity, right? It's the coefficient of dissociation. Like the higher KD, the more it disassociates. To disassociate more, you have low affinity.
Mhm.
Yeah, like Laura's not like a chemistry person. She's not a math person. She's a bio person. Like you have to know these details, but it's not like, oh, I need you to explain the math.
Any other questions?
Like we're wrapping up. It's okay. Like if nobody has any more questions, just wrap it up. Go to bed. No. All right. Yeah. If nobody has any more questions, we can just end the session right here. Yeah, I said no. Yeah, there's no. Well, I hope everyone has a good night. I hope you have a wonderful night's sleep and do as best as you can. Have a good night.
Thanks for coming, guys.
Thank you.